Welcome back. We've been talking about membrane structure and we've moved on to different types of channel and transporter. Last time we talked about uniport, single solute, single direction across the membrane. We talked about symport, two solutes, same direction, and antiport, two solutes, different directions. And I'd mentioned that some of those transporters can actually be using energy to move a solute up a gradient. They can also be used to move things down gradients. So we're going to move our discussion on to talk about active transport. When we were talking about simple diffusion and facilitated diffusion, those are passive mechanisms, and so they don't require an input of energy beyond which exists in a chemical or electrochemical gradient. Active transport, on the other hand, is doing work and therefore requires an input of energy. And now the energy can come in two forms, and this gives us the language of primary active transport and secondary active transport. Primary is pretty straightforward, so let's take a look at that first. Okay, primary active transport is when you're going to have a transport protein like the one here in kind of lilac, light purple color, using the energy that's released when ATP is hydrolyzed to move a solute against a concentration gradient. So here we're looking at a concentration gradient of hydrogen ions, so protons. Remember, a proton or a hydrogen ion is simply a hydrogen atom, which is the nucleus of the protein with a single electron surrounding it. And so a hydrogen ion, a single proton, is simply the nucleus of the hydrogen atom. And so what we have here is 5 protons on the lower side of the membrane, and then I don't know, maybe 10 or 11 on the top side of the membrane. Now I'm using those terms lower and upper or bottom and top, but bear in mind, cells think about things in terms of inside compartments and outside of compartments. They don't have an up, down, left, right, top, bottom. So this will be either inside or outside, and that will be the other side. So let's just say this is inside the cell and that is outside the cell, just for sake of argument here. Okay, so primary active transport will do things like have a transport protein like this one here that grabs a solute, here it's a proton, binds the solute, takes a molecule of ATP, hydrolyzes that molecule of ATP, and then uses the energy released to move the solute up its gradient. And so we're trying to move one of these protons up the gradient. There's very little proton down here and a lot of protons up here. And so that would be primary active transport. It's used in lots of places, and as you get into cell biology and physiology, you're going to start looking at really concrete examples of this. But for now, it's enough for you to know that that's what primary active transport is. Let's compare that with secondary active transport. Now this is a little bit more complicated. Secondary active transport, because it's active, still requires an input of energy, but the input of energy does not come from the hydrolysis of ATP. Instead, it comes from an existing gradient of solute. So what we've got here is a purple symporter, and you can tell it's a symporter because it's moving two different solutes, the white triangle and the purple ball in one direction. So same, two solutes, one direction. So this symporter here is moving the white triangle, which is a proton, same as what we just saw, and this purple ball here is a molecule of sucrose. And so this is something that's happening in plant cells to move sugars in the form of sucrose around the plant body. So what's happening here is that the protons are higher in concentration on this side of the membrane. You can count them up: 1, 2, 3, 4, 5, 6, 7, 8, 9, 10, 11, 12, and 14 protons up here. Obviously in a cell there would be, we're talking thousands, millions of these things. And down here we've got 1, 2, 3, 4, 5 protons. So there's a high concentration of protons up here, low concentration of protons down here. So the protons, there will be a force which will be pulling them down that gradient, and this is also consistent with the idea that the inside of membranes are slightly negatively charged, and so there's actually an electrochemical gradient here. That electrochemical gradient is going to be driving the protons into the cell, and here we're going to allow the proton to move via the transporter, so the protons will come down their concentration gradient. Now, anything that moves down a concentration gradient releases energy as it does so, and that's because when you have a high concentration of solutes on one side of the membrane and a low concentration on the other side, the molecules in the high concentration side, their arrangement and their distribution is a high energy state. It's very much like having water held behind a dam. If you let that water flow down the dam, or through the dam, I guess as it is, you can use that to turn turbines, which can in turn be used to generate electricity. So if we allow the protons to flow down their gradient, there will be a release of energy. And so that energy that's released can be used to do some work. And the work we're going to do here is we're actually going to pump this sucrose molecule up a gradient. And so there's 5 sucrose up here, and now there may be 10 down here. So the sucrose is going from a low region of concentration to a high region of concentration, and that means we're going to have to do some work. That's like pushing a ball uphill. So what we're going to do is let the protons flow down their gradient, release some energy, and use that then to kind of push the sucrose into the cell. This is what happens when you put sucrose into a tissue called phloem, and phloem is what contains the sugary solute that's moved around plant bodies. It's what you tap to get maple syrup out of maple trees, for example. And so proton will bind into the symporter, sucrose will bind into the symporter, there will be a change of shape. Remember, they must both bind simultaneously. There's a change of shape, and the energy that's released from moving the proton down the gradient moves the sucrose up the gradient. So secondary active transport, it uses the energy contained in a gradient to move a solute up a gradient. So where does the first gradient, where does this gradient of protons come from? Well, usually it comes from primary active transport, and so yes, distantly there is a use of ATP, but the proteins that do the movement of a solute in secondary active transport, those proteins are not using ATP as the energy source. So make sure you can compare and contrast primary and secondary transport. So let's put those two things together and look to see what's happening. So is what you've got happening in a cell in plants is this protein here pumps the protons onto this side of the membrane to create a gradient of protons, and then this protein allows the protons to move down the gradient, and as they do so, they release energy, and that can be used to pump sucrose. So you often find primary and secondary active transport working together. Now you look at this and you say to yourself, well, this is stupid. Why on earth would you build it like this? Nobody in their right mind would build two sets of proteins when you could just say, why don't we have the sucrose transporter use ATP? I mean, that's what the proton transporter does, so why doesn't this one do that? That would make more sense. Well, this is the thing about evolution. Evolution doesn't design things so that they are the most well-designed things. It's not about building, an iWatch or a Rolex. It's about building something which works, and this works better than what existed beforehand. And so as far as the plant is concerned, this is great, this is good enough. Evolution deals with just good enough to be better than my ancestor was, so that I can outcompete my ancestor and I can succeed. So people always talk about evolution in terms of its survival of the fittest. You only have to be slightly fitter than your ancestor or a competitor to succeed. And it's actually better to think about it in terms of not survival of the fittest, but death of the weaker, because it's the weak that die and the fitter survive. So yes, this isn't how you design it. That's always going to be the case in biology. Biology doesn't design for goals and intentions. It creates things that work better than what existed in an ancestral generation. So let's look at one concrete example of primary active transport in a physiological context. So some pump proteins, pumps are proteins which will move solutes up gradients. Some of those pumps create a charge difference across the membrane, and so they're called electrogenic pumps, and they're particularly important in nerve cells, in neurons. Now they're important in lots of types of cells, but this is where they're best studied. So there is a protein, I've mentioned this before in passing, there's a protein called the sodium potassium ATPase, and it's this light green protein here, the sodium potassium ATPase. And the sodium potassium ATPase takes the energy that is released when ATP is hydrolyzed, and it moves 2 potassium into the cell and 3 sodium out of the cell. And so the cell in this case would be a neuron. It could be any type of cell, but we'll talk about it in neurons. So this pump uses about 1/3 of your ATP. So if you're a 60 kilogram female student here, you're making about 6 to 7 kilos of ATP, about 1/10 of your body mass every day. You're breaking that down again. Each day. And so 1/3 of that, 2 kilos of your ATP, goes to driving this pump. So the point of this pump is to create a higher sodium concentration and a low potassium concentration outside the cell, and inside the cell the reverse of that, so low sodium, high potassium. And what this actually does is it creates a charge difference across the membrane, and that charge difference becomes important when you look at the function of neurons and you look at action potential generation. So let's take a look in a bit more detail what this protein pump is doing, what this electrogenic pump is doing. Okay, so don't read the slides. Let's see what we got going on here. Okay, so here is the protein to start with. This is called E1. That's the enzyme 1 state. So this is its starting state. And remember, the fact that there are 4 representations of the protein doesn't mean it moves through the membranes. It means this is 1 state. Then in time this happens, and then this happens, and then this happens. So what happens is 3 sodium atoms, 1, 2, 3, they bind into the active site of this transporter. The active site is the part of the enzyme, part of the protein here, where the sodium binds. 3 sodium bind from inside the cell. As soon as that happens, a molecule of ATP is broken down, and 3 of those sodiums are pushed outside the cell. Then 2 potassiums are grabbed. There's a change in shape of the protein again, and the 2 potassiums are flipped inside. Now some of you are probably wondering what this yellow ball is. Well, when the ATP gets broken down, one of the phosphates, ATP remember is adenosine triphosphate, one of the phosphates gets added to the protein, and when that phosphate is released, the 2 potassiums are flipped in. So let's recap. 3 sodiums bind, phosphorylation and a change of shape, 3 sodiums out, 2 potassiums bind, the phosphate leaves, and the 2 potassiums are flipped inside. So this protein is constantly doing this, flipping sodium out, potassium in, sodium out, potassium in. And this generates this gradient of sodium and potassium across the membrane, and that's important in nerve function. So what we've looked at here today, folks, is the difference between primary and secondary active transport. So moving forward, you should be able to compare and contrast simple diffusion, facilitated diffusion, active transport, and then primary and secondary forms of active transport. Okay, that's it for now. We're nearly done with this. We've got 1 short video left to do, and so I'll see you with that right here soon.